All right. So classically, when the professors ask, what's the difference between a prokaryote and a eukaryote gene or genes? So one of them is the one I mentioned earlier, and you guys picked up on it, is the presence of introns. So this one is the eukaryote gene. It has introns. And this is the prokaryote gene. There are actually two genes here. They don't have introns in prokaryotes. And you're like, okay, what is an intron? I don't get it. An intron is a sequence of DNA, or doesn't have to be a DNA, but it's a sequence of, starts in the DNA that's part of the gene. So gene is a DNA sequence that is transcribed. That's the functional definition of it. And so as DNA, it doesn't do anything, but when it gets transcribed, then either it exerts its role as RNA, like ribosome RNA, like tRNA, or has to be translated to protein. That's the case of the mRNA, for it to do something. So the intron is this sequence of DNA that appears in the gene. It gets transcribed. That's why the intron is part of the gene, because it's a sequence that is transcribed. But then it gets called spliced out. So the cell has a mechanism to remove this DNA sequence, or in this case, is mRNA here. It removes this mRNA sequence, and it will translate this, it will translate this and this, but not this. I like to think about it as this is when you're watching the movie without commercials, and this is the commercials, so you splice out the commercials. And people wonder, why do we even have this information in our DNA? It seems like a waste of space, right? And it's kind of like, well, why do you think you have commercials? There is some purpose to it. Bacteria don't have it. They are more condensed in terms of DNA, their genome sizes. I don't know, they don't have time for commercials. They go straight to the movie. Okay, so that's one of the differences. Another difference that I normally ask is about this. It's called polycystronic. So see this one, polycystronic operon. So what happens is this part that is in yellow is a chunk that does not get transcribed. So this yellow part is not a gene. Okay, that's a regulatory sequence, which means it's regulating when this DNA sequence gets transcribed to mRNA. When, how much, how often, how much mRNA should you produce, and it depends on, there are other cues that will bind here to tell you, should you make a bunch of this mRNA or not. So both of them have this yellow sequence here, this and this. They're both regulatory sequence. They don't get transcribed, but they say how much, when, in which tissue to transcribe that gene. Now, that's what they have in common. They both have upstream DNA that is regulating the transcription of the downstream gene. The difference I wanted to point out is that in prokaryotes, you have one regulatory sequence that is regulating the expression of different genes. See that open reading frame here? And how do I know that this gene is different from this gene? See, they're making different proteins. So this could be an enzyme, and this next one is a different enzyme. They don't even have to be variations of the same enzyme. So this is an enzyme, and this is a completely different enzyme. They often are enzymes that are in the same pathway to do something. So classic example in genetics is enzyme to break down sugar. You need like one enzyme, you need another enzyme. So depending on the sugar available in the cell, there will be things binding here to then promote the transcription of these genes, making two different enzymes. In eukaryotes, each one gene has its own regulatory sequence. So we produce one protein out of that one gene. Okay, what I said is true, but not always. And we're like, oh my God, all these exceptions. Yeah, that happens in biology. My husband is a mathematician. It drives him crazy that biology is not so deterministic. You know, it's like, not always. But so as a general rule, each one gene produces one protein. But people found out that in eukaryotes, sometimes that one gene will produce different versions of this one protein. So it's different versions of the protein. Some versions will remove both introns. Other versions will remove just one of them, for example. So the different versions are what happens to the mRNA. It's called alternative splicing. So if you splice both of them, you make this one protein. If there's an alternative splicing, maybe you keep one of these two, then you make another version of this protein. So, but it's still different versions of this protein. It's still considered one gene. So in eukaryotes, each one regulatory sequence encodes one gene. In prokaryotes, one regulatory sequence can regulates the expression of multiple genes. So good? Let's see. Mostly that's what I focus on. There are other differences that even you guys picked up, but what I focus on is more like the structure of the gene, comparing both of them. All right, so good.